So remember, you can like stop and ask me questions, or if you want to type questions in the chat box, you can. You don't have to be on mute. So the last time we talked about the major phases. We talked about the light reactions and the light independent reactions, also called the Kelvin cycle. And then we went through this quiz just now together, and all these answers can be found in the notes. So now we're going to move on to understanding number 6. Temperature, light intensity, and carbon dioxide concentration are possible limiting factors on the rate of photosynthesis. So that means they affect how much photosynthesis happens. So we know that light energy is required for the light dependent reactions. So as you increase the intensity of light, or how bright the light is, you're going to increase the rate of photosynthesis until you hit a certain point. Oops. Let's see. Here we go. Until you hit a point right here. And that is the point at which the chloroplasts are absorbing as much light as they can, and photosynthesis can only proceed so quickly. So at that point, you are flooding the plant with light, and it is processing and absorbing that light as fast as it can. It doesn't matter how much more light you shine on it. Photosynthesis isn't going to keep increasing infinitely. There's only so many chloroplasts. There's only so much surface area in that thylakoid membrane for this reaction to start. So past that point, it doesn't matter how bright the light is. In fact, you'll start burning the plant if you give it too much light. Now, the same thing happens with carbon dioxide. So you can increase the amount of carbon dioxide that's available for the plant to use, but eventually you're going to hit that peak. And at that peak, it doesn't matter how much more carbon dioxide you add, the plant is photosynthesizing as fast as it can. It's similar to the enzyme reactions we learned about in topic 2, right? Where if there's only so many enzymes, it doesn't matter how much substrate you get it. You hit a point of saturation where it can only work so fast. And remember, the molecules that are actually facilitating this experiment are mostly enzymes. So those enzymes are working as fast as they can. Questions? No? Okay. So, the enzymes in photosynthesis have an optimum temperature as well, because like I said, most of the chemical components that are facilitating these chemical reactions in the chloroplast, they're enzymes. They have an optimum temperature and they have an optimum pH. So, temperatures that are too high, right? Here's our peak, here's our optimum. Temperatures that are too high are going to cause the enzymes to denature. And as those enzymes denature, photosynthesis decreases, right? Because they can't do their job anymore. Now, if temperatures are too low, those enzymes aren't going to work either, because there's not enough movement, kinetic energy movement in those molecules for them to run into each other, so the reactions aren't going to proceed. As that temperature warms up, photosynthesis is going to increase until you hit that optimum. This is one of the reasons why, this is the primary reason why plants don't grow in super, super cold temperatures, right? So access to liquid water is one part of it. They need to be able to access liquid water. And the other part of it is that those enzymes just aren't going to work if it's too cold. Questions on that? No? Okay. So now we're going to come to an activity that you're going to do. This is skill number 2, design of experiments to investigate the effect of limiting factors on photosynthesis. And you will have a independent variable and you'll have a dependent variable, and you're going to write the procedure. So we'll get back to this in a minute, there's an activity for you to do in Canvas. I want to go through the rest of the information in the slides, and then we'll do the activity. So just hold on to this thought for a minute. And let's talk about application number 1. So changes to Earth's atmosphere, oceans, and rock deposition due to photosynthesis. So life on land is only possible with plants first, right? Animal life didn't exist on land. Well, aerobic animal life didn't exist on land until plants were here, because Earth's atmosphere initially did not have oxygen in it, at least not enough to support life as you and I know it. So we have a timeline down here. This is going back in time, so 0 is current, 3.8 is going to be, I can't remember if it's million or billion years ago. It's a long time ago. It'll probably show up in a second. Okay. So the first organisms to release oxygen via photosynthesis into the atmosphere were prokaryotes, bacteria that lived in the oceans. So oceanic bacteria starts using respiration, aerobic respiration, and releasing oxygen into the atmosphere. And it wasn't till later that the atmospheric oxygen increased from about 0% to about 2% because of photosynthesis. Now, that increase in oxygen allowed dissolved iron in the oceans to precipitate as iron oxide. So think back to chemistry when you talked about oxidation reactions. So that iron that was dissolved in the oceans can then precipitate out and sink to the bottom. So now you don't have dissolved iron in your water. This is how we get banded iron formations in rocks. And funny enough, we use these bands to track the magnetic changes in the poles. So as new oceanic crust forms, those banding patterns form. And we have basically a banded pattern that shows us which way is north and which way is south, because those iron filings orient themselves to the poles. So every once in a while, you can see the switch of the magnetic North Pole and South Pole. Right now, our magnetic South Pole is up north, and we're due for a shift. We're due for that change to start happening, and we're noticing that the magnetic poles are starting to wobble a little bit. So it's just interesting. And again, we wouldn't have that if we didn't have photosynthesis first to put oxygen into the atmosphere and to put oxygen into the water, causing that iron to precipitate out. Then, pretty recently, if we're talking about in geologic time, that atmospheric oxygen rose to about 30%. And that's due to the evolution of multicellular algae and land plants. And that increases photosynthetic rates, right? That drop back down to about 20%. So, big spike in photosynthetic rates, right? 30% atmospheric oxygen, which tends to be highly reducing. So, that's a little too much oxygen in the atmosphere. And then eventually this photosynthetic rate decreases down to about 20%. So that's how, well, it produces about 20% of the atmosphere, which is oxygen now. So oxygen rates have changed from 0, right? You had prokaryotes that started photosynthesis and raised to about 2%. And then as land plants evolved, you see this big increase in atmospheric oxygen. You hit a max of about 30%, and then you drop back down to about 20% today. Okay, and then these are the review questions for 2.9. So after going through your notes from 2.9, you should be able to do all of these things. So let's go back to our skills activity. So this is the slide. I'm going to bring this. Okay, you can see the Canvas page now, right? Yes, okay. So you're going to click on topic 2. Scroll down to 2.9. And you'll see 2.9, photosynthesis part 2, S2, design and experiment. Go ahead and click on that. And this is what you'll see. So skill number 2, design an experiment to investigate limiting factors on photosynthesis. And there's also a nature of science standard in here. Experimental design controlling relevant variables in photosynthesis and experiments is essential. So your objectives list mechanisms for measuring the rate of photosynthesis and defined independent controlled variables and responding variables. Okay. So your instructions. There is a simulator here. So the simulator is visible here, but for whatever reason, I couldn't get the iframe to show the entire thing, but you can see most of it. So you can work with it in here if you want. You can still, says number of fish, number of plants, light intensity, temperature, and light color. So you can still do that there. Or if you want, you can open the simulator in a new tab. It's right here. It'll take a minute to load. It's waiting. There we go. Okay. So take a few minutes before, I'm going to give you independent time to work on this. So before you get started, take a few minutes and get familiar with the simulator so you understand what it does. And then let's talk about your instructions. So you're going to choose one independent variable. That can be light color, light intensity, temperature, the number of fish. And that's because fish produce CO2. So they will control how much CO2 goes into the system. You're going to need to be able to test this variable at 5 different levels. Okay. You're going to have to write a hypothesis that describes what happens to your dependent variable, which is already set for you. It's the amount of oxygen in the water when you change the independent variable. You need to write a little bit of background that includes the equation for photosynthesis and explain why dissolved oxygen is measured to help understand how much photosynthesis is happening. You're going to have to write a procedure, collect some data. You can recreate this table and complete it. And then you're going to have to write your conclusion, either accept or reject your hypothesis and explain how your data supports that hypothesis or it doesn't. And then you'll need to talk about your controlled variables. Now, you can do this working with a partner that used a different variable than you did, or you can just run the simulation again yourself just to see what happens, right? So that you would be changing 2 variables at a time, and then explaining how that changes your answers. So I'm going to flip over here. Now, this is kind of simplistic, but it's okay. For our purposes, simple is fine. It's actually a little more clear. So we have the number of fish. You can change the number of fish. Now, the number of fish is going to change how much CO2 is available to the plants. So you can change that level. You have to be able to change it to 5 different levels. So 0, 1, 2, 3, 4, 5, right? You can choose the number of plants, but that can't be your independent variable. So you can set the plants for however many you want. I don't care if you have 1 plant or if you have 10 plants. Totally up to you. But that's got to stay the same throughout your entire experiment. You can change the intensity of the light. So you can do 0, 20, 40, 60, 80, 100%. That's fine. That's 5 different levels. You could instead look at temperature. So you can, as long as you come up with 5 different temperatures, you can choose those. You can choose different colors of light. There's 7 here to choose from. You can do 7 different colors of light. Now, what you're doing is you're looking to see how these different variables, whichever one it is that you choose, because you're only choosing one to change, you're going to see how that impacts the amount of dissolved oxygen after 60 minutes. Now, we're doing it after 60 minutes because that's going to be the biggest result. This graph shows you how oxygen concentration changes over time. Part of your experiment is to explain why we are using oxygen as an indication of how much photosynthesis is happening. So, You can submit your experiment a couple of different ways. You can either type it into the text box. If I'm looking at it like you guys, let's see, where's the student vision? Okay, I think I'm in student vision now. Nope, I'm not. Yeah, this is what yours looks like. So you can either, there should be a box where you can type if you hit submit a assignment, I think. You should be able to, yeah, you can either type your answers in there. You can create a 365 document and link it. Or if you want to just write a Word document and upload it, that's fine too. If you want to write it on a sheet of paper and upload it, you can do that too. But I have to be able to read it. So as of right now, that's pretty much all I have for you. If you have questions, you should ask them now. And the rest of the time, you can work as a group. I can leave Zoom up and I can set up a breakout room, or I can just put myself on mute and you guys can work together. Or you can work independently. That's up to you. But for now, go ahead and ask me any questions that you have. And I'll stop recording.